My name is Nina Bauer. I am the EVP for Strategy and Commercial at Fujifilm Cellular Dynamics. In recent years, we have seen an uptick in the use of iPSCs for drug discovery and other testing methods as testing modalities. So we have seen an uptick which was triggered by regulation, number one, but also because iPSC-derived cells are much more human-like because they're derived from humans. So in comparison to animal models, which are traditionally used for drug discovery, they recapitulate human physiology and human tissue in a much more relevant way. So that means if you're testing a new drug on iPSC-derived NAMs or models, you will typically get a much more relevant answer. One big example that we have had with our cells that we make, which are cardiomyocytes or cardiac cells, they are used for cardiac toxicology very often. So that is a later stage. So it's post-discovery checking whether a drug is toxic to the heart tissue and heart function. There's also neurotoxicology, there is liver toxicology. But in the heart model, there was an instance where a drug was developed, it was taken into a phase 1/2 trial, and there were actual patient deaths, which is obviously tragic. The company that was developing these drugs then did a retrospective test to see if this could have been picked up in iPSC models prior to going into human. And lo and behold, they could have forecasted that. So those are moments when there is a realization that having human-like models or human recapitulating models is actually really, really important and becoming more and more relevant and more adopted in the industry. Traditionally, as I say, we've been using animal models. Very often those animal models need to be engineered. So these are gene-edited mice. We also use non-human primates for that. In order to have a colony that is big enough and relevant enough, that actually takes quite a lot of time, whereas cell-based models are readily available off the shelf. So it's also time-based and often much cheaper than having animal colonies. NAMs, or new approach methodologies, is a relatively new term, but that has been around for a while. And the FDA has recently updated their NAMs regulation in such that they now say you can go into human phase 1 based on entirely cell-based models or new approach methodologies. Whereas up until recently, we always mandated animal models. So now we can use cell-based assays. In many cases, those are based on iPSCs. We use organoids or organ-on-a-chip approaches, again, very often based on iPSC-derived cells. And then there's AI methodologies that are being used where large data sets can now be mined and you can kind of forecast safety and efficacy in more of a data-driven manner than an actual in vivo manner on an animal model. If we look at all of these approaches that are now enabled by the regulators, all of them, as I said, are based on iPSCs. So iPSCs in many cases just enable all of these approaches. So that's number one. But also, in many ways, they will ultimately enable the AI models because a lot of the data that will be generated on these iPSC models will then be fed into AI. There is an additional way where CROs and assay developers use iPSC-based cells, derive them into different cell types, be that neurons, glial cells, immune cells, etc. Then they put them together in an organoid, which obviously is much closer to human tissue or a human organ, run that test, and then based on that data, they can then decouple it by taking the individual cell types out and testing them individually to identify exactly what the mode of action is or which cell types are impacted or functional in a particular drug paradigm. So they allow for really detailed analysis of very complex structures by taking individual cells and putting them together into more complex multi-cell type organoids or organs on a chip, or doing the reverse where you use a brain organoid or cardiac organoid, and then once you have the data, you decouple it into individual cell types. So it's actually very, very flexible. So it's a great model system. NAMs and discovery models implies that you have a standardized system, right? So you have the same cell type, you set it up in a particular assay, depending on which question you're asking. Many CROs are now actually setting up these types of assays as an off-the-shelf offering. So a customer can come in and say, I have a new small molecule drug that I want to test for hypertension, whatever it is, and I want to see if it can penetrate heart tissue. Many CROs now have off-the-shelf assays available where they optimized how many cells and which combinations they put on a chip. It really underpins a growing service industry that helps large pharma with their discovery work. Equally, many pharma companies develop these types of approaches in-house where they say, okay, the neuro department will always have this particular neuroassay set up using these cell types. So it is really, it enables almost a commoditization of drug discovery, makes it more efficient, and allows for much higher throughput. And in high throughput screens, you can then much more rapidly scale down to your top 5, top 10 candidates. And so from that perspective, having these standards in place, having a cell source that always comes from the same baseline and can be reproduced and regenerated many, many times so that you can repeat the assay, you can ask a different question with the same assay, but having a slightly different compound. So with these standardized models, you can run large screens with high throughput approaches. You can actually use robotics quite efficiently for that. And you can run many different assays in parallel. So you can parallel path, you can scale down much more quickly, which in theory should be faster and cheaper. iPSC-based models, while highly relevant, still face some challenges in translation, primarily because very often they're just based on 1, 2, or 3 different cell types, which, while called an organoid and similar to an organ, is still not an organ because you're missing the immune system, you're missing blood flow. So there's still a gap between that and an in vivo assessment. I think we have to realize that these models are they are an important step along the way to drug discovery. They will never be the answer for everything. You will still have to either leverage AI based on large volumes of data to predict the functionality of a drug or the efficacy of a drug or the toxicology of a drug. In an ideal world, you will also model in vivo data and in vivo experiences. And as we build AI models with a growing body of data, we should then be able to link everything together and say, okay, if we look at a single cell-based assay versus an organoid versus an in vivo assay in an animal model versus real-world outcomes in a clinical trial, we will become over time much, much better. But I still think we're moving in the right direction with these models. They are not the answer for everything, but they are a very, very valuable tool. Over the past 20 years, and I'll speak from our company experience in this case, we have gone from the first beating heart cells to a plethora of neuronal cells, immune cells, you name it. Like literally you can make any cell in the body. And as people have been working towards these NAMs-based approaches where iPSCs are leveraged to manufacture large quantities of particular cell types for screening and research use only approaches, we have learned, number one, how to differentiate those cells into neurons, cardiac cells, etc., on a routine large-scale basis. We have learned which quality control measures we should leverage and use in order to understand whether our cells are good or not, which helped us in reverse to improve our manufacturing. We have also learned how to freeze these cells down in a manner that we can then thaw them and they're viable and functional. So all of that body of knowledge that we have generated over the years making NAMs directly translates on how we manufacture therapeutics. So any kind of iPSC-based therapeutic that somebody wants to develop, there's probably data and experience that we have or others have from a NAM space that we can directly translate. So as we move through approvals, we've now seen the first iPSC-based therapeutic approvals in Japan. Here in the US, we are seeing moving into late stage for iPSC-based therapies. So right now, there is actually an increasing number of iPSC-based therapeutic developments that are coming through the approval process and moving into first in clinic and then later stage. There's not going to be a cell type that somebody hasn't done, right? So we can leverage all of that body of knowledge and directly translate it to iPSC-based therapeutics and make them in large quantities, make them available, and ultimately make them cheaper, faster, better. And they are off the shelf, they're allogeneic, so they can be readily available for treatments very soon, hopefully. Yeah, this is an interesting one because, as I said, we've been making what is now called NAMs, or more human models, for about 20 years, but it's not until now that there's really a push for a replacement of animal models by these human-based models. I'm not sure what came first. Was it an ask from the regulators to say we need to move away from animal models? Or was it that we're seeing an increasing body of cell types that can now be made in large quantities, reliably and repeatedly at high quality, that the regulators are realizing actually we can move away from animal models? So it's a bit of a chicken and an egg question, and I don't quite know the answer to that. But we're for sure seeing movement both in the US and Europe, and now increasingly in Japan, where it is acceptable not to use animal data or animal-based data. You very much do have to show why your NAMs-based model is relevant and is enough. So you still have the burden of, number one, explaining the context, showing that the data is valid and relatable to whatever the question is, and provide all of the data and underpinning evidence. And it still has to go through the regulators. So they will assess it. They will scrutinize it very much. And there may be cases where they may say it's not good enough. So I'm not sure it really has an impact on regulatory thinking per se, but it definitely has broadened their toolbox and has broadened their general approach on what they feel is acceptable and can be adopted. Yeah. I think advanced therapies for me means many things, number one. So if we want to talk about the next wave of innovation in advanced therapies, we're talking about cell therapies, gene therapies, mRNA-based therapies, or oligonucleotide-based therapies. Probably also talking about ADCs in the widest sense. So there's a large volume of what could qualify as an advanced therapy. We are asking ourselves whether the current paradigm of cell and gene therapy is the one that we will be taking into a longer commercial view, or whether there's still work to do. I think we're at that juncture right now. So there may be a time when people say, well, autologous cell therapy, while great for smaller patient populations, it doesn't scale. But for CAR T, for example, we've seen in vivo. So that is one of the core innovation areas right now. And how can we translate what we have learned in an ex vivo autologous manner into a more off-the-shelf in vivo approach and be much cheaper, much more off-the-shelf, readily available, with much less supply chain issues and shipping things back and forth. The other role that iPSCs can play is that we are now developing much more complex models where we put different cell types together. And so one of those that is actually highly successful in the market right now is a blood-brain barrier model, where one of the big questions is always for neurotherapeutics, can it get into the brain or not? And for the longest time, we thought that larger molecules cannot get across the blood-brain barrier. So we now have an in vitro model where we can recapitulate the composition of the blood-brain barrier in a dish and test that to see whether an AAV or a small molecule drug or an ADC can get across the blood-brain barrier. And I think that is really an area where innovation is happening in a way that we didn't quite foresee, but is really exciting. And we are developing other types of those models. Another one is the neuromuscular junction, to see how a molecule can cross from neurons into muscle, and similar approaches. As we're looking to gene therapy and to the point of how does all of this get influenced by iPSCs, we're seeing a lot of questions being asked around capsids being particularly tissue targeted, i.e. the capsid design is in such a manner that it only infects certain tissues, and so that you can administer it in IV manner, and then it automatically only goes to particular cell types or tissues. So for that, iPSC-derived NAMs are fantastic because you can check all of that in a dish, right? You can grow your target cells. Do they get infected or not? Number one. And then you can grow all the other cells and see if they do not get infected with the AAV of choice. So there's a lot of innovation going into that, doing capsid design so it really only targets particular cell types. We have similar innovation in the mRNA space, where LNPs are designed in such a manner that they target specific cells and not others. Again, same paradigm applies, where you can test that in a dish. Other than that, I think if we're looking at cell therapy specifically, we will be able to learn from our NAMS experience, manufacturing experience, and apply that to large-scale manufacturing in a therapeutic setting, where innovation will be much more on the scalability, cost of goods side of things, automation, implementation, those areas. So yeah, there's a lot of opportunity for innovation. Where particularly it will happen, not quite sure. Maybe on all fronts, hopefully.